bioeducator.com. I'm Dr. Carleen Eaton, and today is the first of 3 lessons on cell reproduction. We're going to start out by talking about the cell cycle. Functions of cell division or cell reproduction include the reproduction of an organism, the growth of an organism, and repair. Certain cells of the body, such as those in the skin or liver, can actually divide. So if you get a cut or an injury, nearby skin cells could divide and help you to heal. The same with the liver. So one function of cell division is repair. As we're going to discuss later on, certain very specialized cells, such as neurons, lose their ability to divide. So they can no longer divide and repair in that way. Another function of cell division is reproduction. For example, bacterial cells divide via binary fission. Binary fission is a means of asexual reproduction, and prokaryotes reproduce via binary fission. Organisms that reproduce sexually create gametes via a particular type of cell division called meiosis. Meiosis creates gametes, such as sperm and egg. Fertilization unites the sperm and egg to create a zygote, which will undergo mitosis, another type of cell division, and thereby grow into an embryo, a fetus, and continue to grow until adulthood. So here we see reproduction. Cells are reproduced via cell division, and growth. So an organism starting out smaller, dividing via mitosis, cell division, and specialization of cell types. So this is just a general overview, and we're going to go into each type of cell division, meiosis, mitosis, and then later on in the course when we talk about prokaryotes, I'll discuss binary fission. So it's important to know some terminology as we go along. When we discuss the cells produced via mitosis and meiosis, these are called daughter cells. And mitosis produces a different number of daughter cells than meiosis. And again, we will go into all of this detail. So remember the term daughter cells. I'll use that throughout the course. Another thing to be aware of is that in order for cells to divide, all of the cell's genetic material must be duplicated. In addition, the cytoplasm, organelles, proteins, and other components of the cell must be replicated. So duplication of DNA has to occur and does occur in one phase of the cell cycle, and additional organelles, cytoplasm, cell membrane are all made to provide enough material for the daughter cells. Right now, we're going to be focusing on eukaryotic cell division. And since eukaryotes have genetic material that is organized into chromosomes, you need to understand chromosomes and chromosome structure in order to understand cell reproduction. So let's take a look at the structure of chromosomes. The genetic material, or DNA, in eukaryotic cells is organized into chromosomes. And this actually shows a chromosome that's already had its DNA replicated. And I can tell that because this chromosome consists of 2 sister chromatids. So this is one sister. This whole section is a sister chromatid. And this is the other chromatid. So sister chromatids. The sister chromatids are connected by what is called a centromere, and the centromere is located here in the middle. So the centromere. And the chromosome overall is composed of chromatin. So if you're just looking at, what is a chromosome made out of? Chromatin. This is DNA plus protein. So the DNA is actually wound around a special type of protein called histones. So histone proteins plus DNA are known as chromatin. At certain points in the cell cycle, the chromatin becomes very highly condensed. And when it's highly condensed, it's visible via light microscopy in the form that you may have seen photos of, which this shows a sketch of, what a typical chromosome looks like. At other points in the cell cycle, it's just chromatin that is less tightly coiled. So again, here we have a chromosome with two sister chromatids. But I want to point out that if you saw something like this, this is still a complete chromosome. So this is one chromosome, and it has only one chromatid on it. This is also considered only one chromosome. It has two chromatids, so it's one chromosome with two chromatids. But these are completely attached and they're identical. So these two chromatids are identical. They contain the same genetic information. So even though this has more chromatids, it's still a single chromosome. And this becomes very important when we discuss the differences between meiosis and mitosis. Understanding that what a chromatid is, what constitutes a chromosome, and how many of these that you have differentiates meiosis and mitosis. So again, you start out with a single chromatid on the chromosome. The DNA is replicated at a particular step in the cell cycle. And you will end up with still one chromosome, but duplicate information. Each section of the chromosome is called an arm. So this is the short arm. So you'll hear referred to the short arm of, say, chromosome 20. So the chromosomes are numbered. In humans, in our somatic, or most of the cells in our body, other than reproductive cells, our somatic cells contain 46 chromosomes. And you hear them called by their number. There's actually two sets, 23 chromosomes, two sets of 23. And this is the long arm and the short arm. So the long arm of a particular chromosome, the short arm. This is the long arm and the short arm of its sister chromatid. With that understanding, let's go on and just talk generally about the outcomes of mitosis and meiosis, and then there will be a lecture covering each of these in great detail. Mitosis results in two identical daughter cells, and it conserves the chromosome number. So, two types of cell division. One is mitosis. And with mitosis, you'll start out with a cell that, let's say, has, for this cell, 4 chromosomes. Mitosis will occur, and you will end up with daughter cells that have the same number of chromosomes. So, conservation of chromosome numbers. In addition, these daughter cells are identical to the parent cells. They're identical in chromosome number. They're also identical in the information that the chromosomes contain. They're identical to each other. They're identical to the parent cell. This is much different than meiosis. Meiosis occurs to produce gametes. So mitosis produces somatic cells. Somatic cells are all the cells in the body that are not reproductive cells. Liver cells are somatic cells. The cells in your eye, your skin, your kidney, those are all somatic cells. They're just the regular cells of the body. Meiosis results in the production of gametes. So gametes are produced via meiosis. So meiosis produces gametes. In other words, sperm and egg. These are sometimes also called germ cells. In meiosis, cells that are going to produce sperm or egg will undergo meiosis, and let's say that a particular cell starts out with 4 chromosomes. With meiosis, there's actually 2 rounds of cell division, and the result will be 1, 2, 3, 4 daughter cells, and each of these will only have half the number of chromosomes. So here we started out with 1, 2, 3, 4. Here I'm just going to end up with 1, 2 chromosomes in each, and these are non-identical. So starting out with 4 chromosomes, ending up with only 2. So the daughter cells are non-identical. They're different from each other, and they are different from the parent cells. Let's look at the cell cycle overall, because mitosis only takes up a small portion of the cell cycle. So there are 2 major phases in the cell cycle, and the cell actually spends 90% of its time in a phase called interphase. So 90% of the time in interphase, and only 10% in the M phase. Mitosis is part of the M phase, and we talk about mitosis a lot because that's where a lot of the interesting stuff happens. That's when the chromosomes are divided into the daughter cells. However, the cell's actually spending most of its time in interphase. So let's first look at the different parts of interphase. There are 3 parts to interphase: G1, S, and G2. G1 stands for gap 1. S phase is synthesis, and G2 is gap 2. During interphase, growth occurs. So interphase is growth of the cell. And if the cell is going to divide into daughter cells, as I mentioned before, not only does it need to duplicate its DNA, it needs to make more cytoplasm, proteins, cell membrane components, mitochondria, Golgi apparatus. And that's what's occurring during these phases. So during G1, there is growth, there is duplication of organelles, increase in cytoplasm, increase in the size and volume of the cell. The cell moves on to S phase, and I want to be clear that the growth continues. The growth is, the duplication of the organelles and everything continues, but something else very important happens as well. Synthesis, S phase, DNA replication occurs. So DNA replication occurs so that there will be enough DNA for each of the daughter cells. After S phase, the cell goes into another gap phase. This one's called G2 gap 2, and during this time there is continued growth. The cell will again duplicate organelles, continue to increase in size, and then by the end of gap 2, the cell has everything it needs to produce daughter cells. So this is all interphase, and now we're in the M phase. There are 2 portions of M phase: mitosis and cytokinesis. Mitosis is the section of the cell cycle during which the chromosomes are divided up into 2 separate sets, 1 for each nuclei of the daughter cell. So here we have separation of chromosomes. Division of the chromosomes are divided up into 2 pairs, or not 2 pairs, 2 complete sets, 1 for each nuclei of the daughter cells. Now, cytokinesis is the actual separation of what was 1 cell into 2 cells. So now we have 2 sets of chromosomes, everything we need for 2 cells, but it's still just 1 cell. The cell pinches off in the middle, the cytoplasm divides, and you end up with 2 daughter cells. Cytokinesis is the actual physical separation into 2 cells. Cytokinesis actually starts during late mitosis. It's sometimes considered a part of mitosis or telophase. Sometimes it's written as an extra step afterwards. Either way, it's part of M phase. And you have to remember these divisions into phases, mitosis versus cytokinesis is arbitrary. It's something scientists use to help them discuss and categorize and study the cell cycle. But in nature, things are more continuous and there's not these absolute separations. So mitosis and cytokinesis, there is some overlap in these processes. mentioned before that certain cells, such as neurons, don't divide. Very highly specialized cells don't divide. So what do? Where are they in the cell cycle? Well, they're arrested in a quiescent phase known as G0. G0 is just a resting state in which the cells don't proceed through the cell cycle. The cells that are in this state are sometimes called post-mitotic cells because G0, that phase, is after mitosis. Sometimes G0 is considered just an extension of the G1. Other times you'll just see it written here as just immediately post-mitosis. Either way, G0 is a quiescent resting phase that non-dividing cells stay in. Let's focus a little bit more on cytokinesis. Again, after the cell has duplicated its organelles, duplicated its DNA, and then gone through mitosis, what will happen is you will have this one cell that's now larger than it started out with, and two sets of chromosomes, one for what's going to be the nuclei of each daughter cell. But now these cells need to separate. This cell needs to separate into two daughter cells. And it does that in animals through what's called a cleavage furrow. So a cleavage furrow forms down the middle of the cell, and that's this indentation that you see here, this groove, is the cleavage furrow. Actin and myosin pinch off the cell in the middle. Remember that actin and myosin are proteins that are also important for the contraction of muscle and for different kinds of movements. One of their functions is to interact in such a way that this cleavage furrow deepens and pinches off, and then you end up with two physically separate daughter cells. 1, 2, 3, 4. 1, 2, 3, 4. And as you'll see here, these there are 4 chromosomes, but each only has one chromatid on them, and we'll see why in the mitosis lecture. Now, plant cells function a little bit differently as far as cell division, and that's because they have cell walls. So instead of forming a cleavage furrow, what happens with plants is that they form what's called a cell plate. In telophase, vesicles that are formed by the Golgi apparatus start to coalesce near the middle of the cell. So these vesicles will coalesce, and what they contain are components to build a cell wall. So this cell plate then forms, and a cell wall can be built between what are going to be the two daughter cells. And then those will divide into two daughter cells, but they stay closely associated with each other in plants, the two daughter cells, and they're actually stuck together. So plant cells, the division is a little bit different. Again, it involves a cell plate versus a cleavage furrow. One very important topic is that of control of the cell cycle, because different cells divide at different rates. Certain cells, like skin, are rapidly dividing. They'll go through the cell cycle a couple times in one day even. They have a rapid turnover. Other cells, like neurons, are arrested in one phase of the cell cycle. And if the cell cycle is not tightly controlled, the result will be out of control growth and sometimes cancer. So let's talk about how the cell cycle is regulated. There are cytoplasmic signaling molecules that regulate the cell cycle, and there are also other internal and external factors that regulate the cell cycle. First, focusing on just these cytoplasmic signaling molecules and what's called checkpoints. So these molecules constitute what you'll hear called a cell cycle control system. Its name tells you what it does, controls or regulates the cell cycle. There are checkpoints in the cell cycle, and these occur at G1, G2, and M phase. Checkpoints are points where the cell stops and waits for a go-ahead signal. When you look at the cell cycle, that schematic of it, you see that, there's G1, S, G2, and you might think that, okay, the cell just moves, it does G1, it finishes, it goes on to S phase, then goes straight to G2. But that's not actually the way it happens. the cell does not automatically move from one phase to the next phase. In fact, it stops after a certain phase, it waits. If it gets a go-ahead signal, it goes to the next phase. If it doesn't, it stays there. You can think of this as like a stoplight. At these different checkpoints, the cell gets to the checkpoint, the light is red. It waits for a specific signal that is analogous to a green light. When it gets that signal, it goes to the next phase. The most important checkpoint in mammalian cells is the G1 checkpoint. This is often called the restriction point. If a cell receives the go-ahead signal in the form of these cytoplasmic signaling molecules, the cell will generally advance on through G1, S phase, G2, M phase, and end up dividing into two daughter cells. So it's very likely, if the cell gets the go-ahead at this point, that it will complete the cell cycle. If no go-ahead is given at this restriction point, the cell will stay in G0 quiescent phase and remain there. All right, so what are these cytoplasmic signaling molecules? We know that at least some of them are cyclins and cyclin-dependent kinases. Recall in a previous lecture, I mentioned that kinases are proteins that phosphorylate other proteins. So kinases phosphorylate other proteins. In doing so, these kinases can either activate or deactivate another protein. CDKs, cyclin-dependent kinases, the name tells you something about them. They're dependent on cyclins. So CDKs are cytoplasmic proteins that are inactive until they are bound to cyclin. Active CDKs give the go-ahead signal at the restriction point and the other checkpoints. So these CDKs are existing in the cytoplasm, and when the cyclin interacts with the CDK becomes activated, and when that occurs, it can tell the cell, go ahead to the next part of the cycle. Scientists have done experiments in which they've determined that when the level or the concentration of cyclin increases in the cell, CDK activity also increases. So increased cyclin concentration, the result is increased CDK activity, which makes sense if cyclins are necessary to put CDKs in their active form. When the concentration of cyclins decreases, CDK activity decreases as well. Okay. There are other internal and external factors as well that regulate the cell cycle. To point out also, the first CDK that was discovered that you should actually know the name of is MPF, and this is known as M-phase promoting factor. This is the go-ahead signal, gives the go-ahead at the G2 checkpoint. Now, CDKs and cyclins are necessary for the cell to go through the cell cycle, but they are not sufficient. There are other factors as well. So even if there's the go-ahead, if there's some other factor that is not correct, the cell may not continue to divide. Internal factors. Let me give you an example of an internal factor. An example of an internal factor would be chromosomes lining up, being all lined up correctly at a certain part of mitosis. So even if the go-ahead signal was given, everything the cell needs to divide is there, if the chromosomes aren't in place, it's not just going to keep going. So internal factors, making sure that everything's in place, in the correct order, before the cell goes on. So example of an internal factor is cell waits for chromosomes to line up before proceeding with mitosis or meiosis. That's an internal factor, something inside the cell. CDKs are internal as well, but they're put in a slightly different class. Okay, a second type of factor, external factors. These are things that come from outside the cell. For example, nutrients. So even if the cell gets the go-ahead signal, if it's in a situation where there's not nutrients, that's probably not a good time to divide because there won't be enough nutrients to support the daughter cells. Growth factors. Growth factors are secreted by cells and they induce other cells to divide. So they don't come from within the cell that's going to divide, they come from outside it. So one cell secretes a growth factor. This cell secretes a growth factor, and what that's going to do, that growth factor, is act on nearby cells and tell them, okay, so this is the growth factor, let's say, and it's going to tell this cell over here, divide. So that's an external factor. So these are factors that tell the cell it's okay to divide. The go-ahead signal is given, the internal factors are in place, the external factors are in place, go ahead and divide. Conversely, there are factors which stop cell division. So the cell knows that it needs to stop dividing under certain conditions. We talked about if, maybe there's not enough nutrients or the chromosomes aren't lined up, but there's other things as well. And two important facts to remember is that there is something called contact inhibition and anchorage dependence. Contact inhibition means that the cell will not divide if the situation is too crowded. So it's inhibited by contact with too many other cells around it. This is also called density-dependent inhibition, and this exists both in the lab, in vitro, and in a living organism, in vivo. So if you are working in a lab with cell culture, you'll see that the cells will just stop dividing once they reach a certain density. They won't just pile up and pile up, and that's due to contact inhibition. Anchorage dependence means that a cell needs to be attached to a substrate. In the lab, if you're growing cells, the container that you're growing the cells in, the cells can attach to. And cells floating around will not just be floating around and dividing. They have to be attached. So normal cells exhibit contact inhibition and anchorage dependence, meaning that they will not divide if the situation's too crowded, and they won't divide if they're not anchored to something. That's normal cells. Cancer cells lose these controls. So let's talk about cancer cells. First, cancer cells may not necessarily wait for the go-ahead signal at the checkpoints. So I talked about the go-ahead signal. They might just plow through and go through the cell cycle and go through it again and not wait to get the go-ahead. So that's one way in which cancer cycles don't adhere to the normal regulation of the cell. As I mentioned, they also exhibit a loss of density-dependent inhibition, or this is the same as contact inhibition. So even though they're in a really crowded condition, they'll just keep growing. They also lose anchorage dependence, meaning that they don't need to be attached. They might grow even though they're not attached to a substrate. The result of these various factors is that the cells can form tumors. And the cancer, as you know, can also spread. It can spread to nearby sites. It can also spread to distant sites. And this spread to distant sites of the body is called metastasizing, or the cell metastasizes. So metastases spread to a distant site. Another thing that cancer cells do is they actually induce blood vessels to grow towards them. So a tumor will actually somehow induce blood vessels to grow toward it. When that happens, now those cells have a means to get to other areas of the body. And cancer cells actually use vessels, blood vessels, and lymphatic vessels to spread. Then, since they don't exhibit anchorage dependence, they don't have to be on a substrate, they'll start growing somewhere else in the body, and even if things get crowded, they'll still grow. And then they can spread to areas and damage them. For example, let's say that an individual has breast cancer. She has a tumor in her breast. Some of those cells from the breast cancer can invade bone. So they spread to the bone, they grow there, and can damage the bone and cause fractures. So when scientists are studying cancer, one thing that they focus on is how are cancer cells different than normal cells, and here's some ways in which they're different. And later on, when we talk about molecular biology, we'll look a little more closely at the molecular level about regulation of genes and how that can affect cancer. All right, let's do a few examples to reinforce what we've just learned. First of all, example 1, label the parts of the chromosome below. What is the complex of DNA and protein of which chromosomes are made called? What is the protein component of this material called? So parts of the chromosome. Well, first of all, I see that there are 2 sister chromatids here, and each of them has a short arm, and each of them has a long arm. And there are sister chromatids, so we can label those sister chromatids. Remember that sister chromatids are connected by what's called a centromere. So we have the short arm, the long arm, sister chromatids attached by a centromere. What is the complex of DNA and protein of which chromosomes are made called? So what is a chromosome made out of? It's made out of chromatin. What is the protein component of this material called? Remember, chromatin is made of DNA and protein. What is this protein? It is histone protein. So histone proteins plus DNA equals chromatin, and that is what a chromosome is composed of. Example 2, the figure below shows a schematic representation of the cell cycle with the G1 phase labeled. Label the remaining phases. What phase are non-dividing cells arrested in? So starting out with the G1 phase, which is a growth phase, it's gap 1. Cells that get the go-ahead will continue on into the S phase. During the S phase, growth continues, and in addition, DNA is replicated. So it's the synthesis phase, S phase for synthesis. The next phase is G2. This is a second gap phase, gap 2, and during this phase, growth continues. All of these 3 together are known as interphase. Remember, the cycle spends the vast majority of its time in interphase, about 90% of the time. Then the other 10% of the time is spent in M phase, the mitotic phase. Mitotic phase consists of mitosis, which is the separation of the chromosomes into 2 groups, 1 for each of the daughter cell nuclei. That is mitosis. Cytokinesis is the physical division of the parent cell into 2 daughter cells. And remember, in animal cells, that's via a cleavage furrow. In plant cells, it's via the formation of a cell plate. So these are the phases. Now, what phase are non-dividing cells arrested in? These cells are in the G0 phase, a quiescent phase, and they are called sometimes post-mitotic cells. Where are the checkpoints in the cell cycle? There are 3, G1, S, and G2. And remember that G1 is called the restriction point. And if the cell gets to go ahead at G1, there's an excellent chance it's going to make it through the whole cell cycle. What are 3 means by which the cell cycle is regulated? Well, the first one is cytoplasmic factors. And specifically, these cytoplasmic factors are cyclin and cyclin-dependent kinases, or CDKs, cyclin-dependent kinases. So that is one way in which the cell cycle is regulated. It's also regulated by internal factors. And the example I gave was the chromosomes lining up. So everything has to be in place, in the correct location for the cell cycle to continue. The third type of factors is external factors. External factors include things like nutrients or growth factors, the presence of growth factors. Remember that there are also ways in which the cell cycle can be stopped, that it can be stopped by contact inhibition when things become too crowded. It can be stopped by anchorage dependence. So if there's nothing for the cell to hold on to, there's no substrate, it won't continue to divide. Example 4, how does the loss of contact inhibition and anchorage dependence allow cancer cells to spread to other areas of the body? Okay, contact inhibition, I just mentioned, should stop a cell from dividing. So if things become too crowded conditions, normal cells stop dividing. Cancer cells do not. Cancer cells can keep dividing in these conditions. Cancer cells may keep dividing. This allows them to form tumors. So even though it's crowded conditions, cell continues to divide. The second one is anchorage dependence. If there is no substrate for the cell to hold on to, normal cells stop dividing. Cancer cells don't. This allows them to break off, go through the bloodstream, travel or metastasize, form metastases, and grow in distant sites of the body. So they don't need a substrate. They can travel through the bloodstream or the lymphatic vessels, set up elsewhere, and divide. And form tumors and continue to divide, even though it's very crowded to the point that it's damaging the organ. So the lack of contact inhibition and anchorage dependence allows cancer cells to spread to other areas of the body, metastasize, and form tumors. In addition, one thing to note is that a normal cell will only divide a set number of times. It'll only go through the cell cycle, let's say 50 times, and then it will die. Cancer cells sometimes become immortal. They will divide and divide and divide indefinitely. So we call that immortalizing. And that's another reason that tumors form and cancer spreads is they have an infinite number or a very high number of cell divisions that they can go through, unlike normal cells. So that concludes this lesson on the cell cycle. I'll see you again soon here at educator.com.